Thanks, Rob. And I want to thank the organizers as well for the invitation. I was originally slated to talk about the evolutionary emergence of infectious diseases and things like drug resistance as an example of biological invasions, but I emailed the organizers because I've been working on some general theoretical ideas about quantifying constraints in multivariate phenotypes, and they kindly allowed me to change the title of my talk and the subject of my talk. Hopefully I won't delve too deeply into the obscure and weird things of theory, but I want to convince you that there are some interesting aspects of what I'm going to show you in information theory that can be used to quantify genetic constraints in a way that hopefully you'll see is relevant for things like invasion biology. I should also say before I begin, this is joint work with Fadi Al-Ajaji, who's a faculty member in my department, and three really excellent second-year summer students that we've had this summer that have been exploring these things. So Mark gave a fantastic talk that sort of sets a lot of the stage for what I want to talk about, and Rob also gave a fantastic talk that sets part of the stage for the other things I want to talk about. And so I have here just a figure stolen from a paper by Rob and Spencer to kind of motivate what I'm going to talk about in the context of invasion biology. And so you've already heard Rob talk about this purple loosestrife story, and he showed you evidence that if you look at two traits, say like time to first flower and size at flowering, there is selection that's in a direction that seems to be not in the direction where most of the genetic variance is. And so in terms of an invasive species, if this is the thing you need to be as an invasive species, there's obviously a genetic constraint here, and that genetic constraint can have important roles in terms of whether or not the species will actually be a successful invader. What I want to do is consider how we go about quantifying genetic constraints like that. So there's the motivation, and I'm going to delve into the details now of quantifying genetic constraints, and I'm going to take off basically from where Mark started. So how do people typically imagine quantifying these sorts of genetic constraints? Well, the first thing I want to highlight, which has already been mentioned a few times, is that the focus is typically on standing variation. And it's kind of hard to imagine how you do anything other than that, short of knowing something a lot more about the developmental biology of the species in question and how mutations affect that. Then we take the multivariate breeders equation as a background for how we want to think about selection acting, and that decomposes the response to selection into a genetic component, this G matrix that Mark talked a lot about, and beta, this selection gradient. And we want to understand the genetic constraints involved, and that amounts to analyzing the G matrix like Mark talked about. And that means analyzing what you'd call the eigenstructure of that matrix. And as Mark explained, you want to know what the eigenvectors are and what the eigenvalues are. Now, in the context of a bivariate plot like that, this is a principal component analysis, basically. So these things are nothing more than the first eigenvector is going to give you the axis of major variation in the plot, and then the second eigenvector is going to be a vector orthogonal to that. And so we can think of these as PC1 and PC2 as the two eigenvectors, and the eigenvalues associated with those eigenvectors are measures of variation in those different directions. There's a big eigenvalue associated with that, and a smaller one associated with that. Now, what I want to focus on is the fact that this kind of an analysis is fundamentally tied to an assumption of multivariate normality. And I want to be specific about what I mean by that. You can conduct a principal component analysis on any data. That's not specific to multivariate normal data. And in fact, the breeder's equation here is perfectly fine as an equation, at least for one generation, to predict the mean after one generation of selection. But when we use this sort of analysis of the eigenstructure of G to understand genetic constraints, we're all of a sudden making a very important assumption about the fact that these data must be multivariate normal. And I want to explain exactly what I mean by that. So here's something else taken from a paper by Rob and Spencer, which I'll return to in a second. But first, I want to focus on what it means when the data is multivariate normal in a bivariate case. And in that case, if we think of the level curves of the probability density, the level curves of the probability density of a bivariate normal are going to be ellipses. And so when we conduct our principal component analysis on that data, we're going to get the first eigenvector and the biggest eigenvalue associated with the major axis of the ellipse of that distribution. And then the second one, what it's going to do is it's going to pick out an orthogonal direction to that, the next biggest amount of variation, orthogonal to that if there's more than two dimensions. And what we'd like to know is, where are there genetic constraints? Are there directions where there's very little genetic variance? And as Mark showed you in what he was looking at in his data, one way to do that is to focus on the eigenvalue associated with the smallest eigenvalue, basically. If that smallest eigenvalue is 0, then in some sense you might expect that there would be 0 variance in some direction in this multivariate space. And that's perfectly fine when it's multivariate normal because these level curves are ellipses. And you could imagine if I rotated this smallest axis here in any direction other than the one it's currently pointing in is going to give me even more variation. So that really does pick out the direction of smallest variation. But once I'm in a world where things aren't multivariate normal, so imagine a kite-shaped distribution like this, and I pull this out only as motivation for the fact that, these kinds of things are not unreasonable expectations in some data sets. We can again do our principal component analysis, find our eigenvectors, our eigenvalues. This axis is no longer the direction of smallest variation. So if I look at the eigenvalues to try and understand where the genetic constraints are, I no longer get the right answer. There are other directions that have smaller variation than the smallest, the eigenvector associated with the smallest eigenvalue. And in particular, in this plot, there's a direction in that orientation that has smaller genetic variation. So what this means is if the data are not multivariate normal, and you look at the eigenstructure of G and you find lots of variance, big eigenvalues, that doesn't mean that there's genetic variation in all the different dimensions. There can be directions in which there is no variation, but you wouldn't see that in the eigenvalue structure of the matrix. Interestingly, the opposite is also true. So you could imagine a probability distribution of breeding values, say, that has some kind of level curves that look like that, so maybe a bimodal distribution. Perfectly fine. You run a principal component analysis on G again. You pick out this is the direction of greatest genetic variance, and your analysis will tell you that there's no genetic variance in the other direction. And so you might mistakenly, in this case, conclude that there's only one trait here, because the other eigenvalue associated with this eigenvector is going to be 0. And you can see that's not true. There's no variation in that very specific direction, but any other direction I choose, there's quite a lot of genetic variation. And so it would be wrong to conclude that there's only a one-dimensional trait space here, because clearly there are places I can move where I can evolve both traits to some extent independently. And again, this is all due to the fact that it's not necessarily multivariate normal. Now, of course, this kind of thing, in a sense, wouldn't be a problem if we were only interested in bivariate plots. And because if we were interested in bivariate plots, we could just plot the data. We could look at it and see if something weird is going on like that. If it is, maybe we could transform the data to make it multivariate normal. Even when it isn't multivariate normal, we would at least be aware of the problem and we wouldn't make any of these sort of interpretive mistakes. But really, in many of these instances, and this is again data from Rob and Spencer, people are interested in a lot of traits, I think 12 or 13 or something like that in this case. And it's no longer possible to visualize that. So you can't visualize the data anymore to determine whether there's something weird going on. One thing you could do is test for multivariate normality. A lot of the time those tests have pretty poor power when you're dealing with large dimensional data like this. I'll show you another set of data that Mark kindly provided to me that he already talked about in his presentation previously. These are these 8 traits, these 8 CHC traits that he talked about in his Drosophila experiment. All of these marginal distributions look sort of normal-ish. If you run a univariate normality test on these, you find that some of them aren't actually normal. If you plot the bivariate plot of these different things, you can again see, at least in certain plots like 1 and 2 here, that again, it's got a strong correlation structure, but it's clearly not multivariate normal in that there's a high density here and the skew that goes off in the direction of this correlation. Again, you might transform that data if it was only the bivariate stuff, but it's hard to know what's going on in the 8-dimensional space. And so my interest in thinking about this was to try and see if there were ways you might try and capture the dimensionality of space, of the genetic space in a sense, without restricting attention to the multivariate normal kinds of examples. And so the way I want to do this is I want to step back for a moment and consider how you might measure evolvability and constraint in a univariate case. And once I develop an idea for that, I'm going to scale it up to a multivariate setting. So the idea here is and I'm going to focus on breeding values. So we've got a distribution of breeding values in generation T, and here it is in generation T plus 1. And I want to measure how much evolvability there is in this population. And the way I'm going to do that is I'm going to think of 2 things. First, I want to ask how much evolution has happened in moving from here to here. I want to quantify that in some way without assuming any particular distribution for the breeding values. And then I want to quantify how strong selection was to generate that change. And I'm going to use this ratio, the amount of evolution divided by the strength of selection, as a measure of how evolvable a population is. So if that's really big, that means I can get a lot of evolutionary change with very little selection. If it's small, I have a hard time getting any evolutionary change, even with really strong selection. So that's what I want to use as my measure of evolvability. Of course, the inverse of that is a measure of constraint. And so I need to come up with a general way of quantifying those two things. And so let's first think about how much evolution has happened. So here's a population at time t. In most of the cases that I've seen where people are interested in these sorts of things, the primary focus is on understanding evolution as a result of selection. So in other words, not so much in terms of things like drift or other forces that might cause evolution. And so really what you can think of in terms of what's happening evolutionarily is nothing more than bias sampling from this distribution. And the bias is a bias that's determined by the fitness of the different breeding values. And so I can somehow quantify how much evolution has happened by quantifying how much biased sampling there's been, is the idea. And so the way I want to imagine doing that is, let's suppose we took an unbiased sample from this distribution. Really, it's a biased sample, but let's imagine taking an unbiased sample of size n. We could then ask, what's the likelihood of actually getting a population that looks like this if the sampling was unbiased? If that's really low, then in some sense, there's been a lot of evolution that's happened here because there's been a lot of bias in how the sampling has occurred. If that's very high, then there hasn't been much bias, and so the distributions are in some sense very similar, and not much evolution has happened. And so I want to quantify that. I want to do it for large samples, though, because I don't want to allow for anything like genetic drift to happen. So these things, and I'm going to go through a bit of the mathematical detail only because what I end up with will seem kind of mystical if I don't. So I want to show you where it comes from so you can appreciate, if you're interested, where the mysticism lies. So it's easy to quantify that likelihood. If I take a sample of size n and I want to know the likelihood of getting this, well, I need to get C sub I samples from bin I, and the probability or the likelihood is just a multinomial probability. And then I want to let n get big because I want to allow this to work for big sample sizes. Of course, this doesn't behave well when n gets big, it gets really small. So I want a measure per sample in some way. So I want to standardize by sample size. I want to divide by sample size, essentially. And to do that, I'm first going to work with the log likelihood just because it has better properties and because it's tied to some nice statistical ideas as well. So there's the log likelihood, and I'm going to standardize by n. And then I want the sample size to be big so I don't have to worry about genetic drift. And if you do that, you end up with this, what turns out to be a really nice quantity here that involves the probability distribution here and the probability distribution there. And the reason I say it's really nice is because this is a quantity that's been around for quite a long time in the information theory literature, surprisingly enough. And it's called the Kullback-Leibler divergence. And it's a measure of how different two probability distributions are. Roughly speaking, what it tells you is how much information, in a sort of precise sense that I'm not going to go into here, how much information would I lose if I use this distribution here when really this is the true distribution? So if that's a lot of information that I lose, then those distributions are very different, and if it's zero, then those distributions are the same. So that's what I'm going to use as my measure of the amount of evolution that happens in one time step. Then I need a measure of the strength of selection. And of course, the way that is going to be formulated will be determined by a model of selection that I choose. And I'm going to choose a model that's, again, kind of motivated by this stuff that Rob talked about, where, you know, in this context, really what's of interest here is the fact that there's a hole here in genotype space. There aren't genotypes that are the ones that you'd want to be the good invaders. And so I'm going to imagine, rather than imposing something like directional selection or Gaussian selection, I'm going to select for a particular thing, like this particular spot in a genotype space. Now, in the context of a univariate situation, that means I'm going to select for a particular bin, a particular breeding value. Now, that bin can have different widths, and I'm going to refer to that as the resolution of selection, because I might be focusing very tightly on a small range of breeding values, or I might have a reasonably wide bin. And so I want to then use that model of selection to come up with a quantity that is the strength of selection. And what I'm going to use is basically the analog of the selection gradient in this setting. So that's this quantity here. This is basically the analog of the gradient of the mean fitness of the population. So that's, in one way at least, a natural measure of how strong selection has been. And that's for a particular k, and of course I want to allow k to be any value in order to get a kind of general sense, a general measure of evolvability without specifying what specific trait I'm actually interested in. Well, so now I look at the ratio of those two things, this measure of divergence for the amount of evolution and this measure of the strength of selection, and surprisingly enough, it comes out to a really simple formula for a particular place that I put my fitness function k. And then I want to average that, or actually what I'll do is I'll sum that over all the different places I could have selected, all of the different traits I could have selected. And this comes out to a really nice quantity that, again, is found in information theory. It's Shannon information, or it's the information entropy of the probability distribution, or the information entropy of the breeding value distribution in this case. Now that's, I'm sure, this is why I wanted to show you where it came from, because it's a mystical thing that crazy people talk about sometimes in evolutionary biology. I want to give you a feel for what it is, just because it's probably not familiar to everybody. And in a way, it's a measure of uncertainty. So if I think of the space of different breeding values as a bunch of different bins here, I can spread that probability mass over those bins in a bunch of different ways. If it's really concentrated, so it's only in a single bin here, then there's very high certainty. It's very ordered. There's very low entropy. If I spread it over more bins, then it's very disordered. The probability mass is spread over all different places in the different values of the breeding value. It's high entropy. There's a low level of certainty in terms of what would happen if I sampled from that distribution. The reason it's used in information theory is because another way you can think of it is how much information I gain by witnessing a realization of a draw from that distribution. If I had a draw from this distribution here, I wouldn't really learn anything because I already know, given I know the distribution, I already know exactly what's going to happen. There's probability 1 that this event is going to happen. Whereas I learn a lot more here because there's quite a bit of uncertainty until I witness what happens, what the possibilities are. Maybe a little bit more naturally in evolutionary biology, it's also a measure of what I'll call variability as opposed to variance. So here are two distributions, and there is an important sense in which both of those distributions have the same variability. There are two things that can happen and they're equally likely. It doesn't matter where they are here, there are just two things, they're equally likely, and so there's the same amount of variability in a certain sense. Of course, the variance is very different. The variance here is big and the variance here is small. These two things have the same information entropy, but obviously they differ in variance. And so in terms of a measure of evolvability, this information entropy quantity would characterize both of these things as having the same amount of evolvability, whereas a variance measure would say this one has a greater evolvability. So in a sense, I think that this is just to argue that you can go through those calculations, that's all fine and good, but at the end of the day, it ought to make some degree of intuitive sense too. And I think that at least to me, it's reasonable to imagine both of these things have the same amount of evolvability. Here's another scenario, again, not normal, obviously. This is a population that has a reasonably high variance, and this one has low variance, because all I've done is chopped the probability mass and filled in the hole in the middle. This one has a low entropy, and this one has a high entropy. So my measure of evolvability would say that this has a low evolvability, whereas a variance measure would say it has a high evolvability, and I think the entropy measure there better captures the fact that there isn't much you could do evolutionarily with this, whereas this has a high evolvability. So again, just a couple of arguments for why this is maybe a reasonable measure of evolvability in the case of non-normal populations. And just to make it even more believable as an intuitively sensible thing, well, I won't talk about this too much, but it ties in nice ways to Bayesian statistics in terms of how people quantify constraint in distributions. And information theory is used as a way to quantify constraint. But in the case of normality, in the univariate case, the information entropy reduces to a quantity that's really nothing more than a function of the genetic variance. So it reduces to what you'd hope it would reduce to in the case of normality. And in multivariate normality, it reduces to something that depends on all the eigenvalues of G, which is what Mark talked about. So again, you'd hope that a generalization in some way reduces to what you'd be using in the case of multivariate normality. Now you want to scale this up to multivariate settings, and that's actually really easy to do in the context of information theory. I have a trait X, I have a trait Y. I can calculate the information entropy for both of those traits, and I can calculate the joint entropy. I'm using a Venn diagram because these information characteristics have the nice property that they're additive. They're a perfect measure of dependence, not necessarily just covariance or correlation, but any kind of statistical dependence. And the overlap between these things represents mutual information between those two variables. If there's no overlap, they're two independent random variables. If there's perfect overlap, there's really only one random variable there. And if there's partial overlap, then there's some degree of dependence between the two. And I'll just run through, so one thing you might naturally do then as a measure of dimension, or the number of dimensions in a data set, is you might take the joint entropy, which is the union here, and you might divide by the sum of the marginal entropies. So that's going to give you a fraction of, so if they're independent, they're completely disjoint, and so the joint entropy is going to be the sum of the areas of those two things. But if they overlap, then that union is going to be slightly smaller than the amount of entropy you could have had. And so this will be some fraction of the total possible entropy when the traits are really independent. You multiply that by the number of traits, and that gives you a measure of dimension, taking into account basically any kind of statistical dependence, regardless of what the distribution is. And I'll skip over that and just give you a couple of examples. So here's a 2D example where I'm doing that. So here's a case where we have two traits. Let's imagine this is the distribution of breeding values. There's no significant correlation or covariance to speak of in this data, but there's a pretty clear pattern of dependence here. If I know trait 1, I know an awful lot about trait 2. If trait 1 is low, then trait 2 is also low. If trait 1 is high, then again, trait 2 is typically low. But if trait 1 is somewhere in the middle, then trait 2 is high. So there's a clear dependence between these two things. It's not multivariate normal. And I'd like to be able to pick out the fact that there aren't really two dimensions here. There's more than one. There's somewhere in between. And so what you can do, as I said, is you can look at those marginal entropies and you can calculate that quantity that I showed you. And it comes out to 1.67 in this case. So there's a degree to which these two traits are dependent. There's not two dimensions here to play with, but there's more than one. There's somewhere in between. As another example, you could imagine, so here's another case where there's a functional dependence between two traits. So there, again, the covariance is really not much to speak of here, but you can see if I know trait 1, I know everything about trait 2, because trait 2 is just a nice function of trait 1, some nonlinear function of trait 1. And so I'd like to be able to pick out the fact that there aren't two traits here either. In some sense, there's maybe only one trait. Well, again, I can look at the marginal distributions of those two traits, and I can calculate this information entropy for those two marginal distributions, and I can quantify how many dimensions are in this data set. What you'll notice here is that the marginal entropy of trait 1 is exactly the same as the joint entropy of the whole distribution, and that's because if I know trait 1, I know everything. The marginal entropy of trait 2 is not quite the total joint entropy because trait 1 is not a function of trait 2. Trait 2 is a function of trait 1, but the opposite is not true. So knowing trait 2 tells me a lot about trait 1, but it doesn't tell me everything. So there's not two traits, but there's not one either according to this measure. There's a little bit more than one dimension in this particular genotype space. So that's just hopefully to give you a flavor for the idea, where it comes from and how it would work with some kind of made-up distributions. What I want to do now is, so I took the data Mark sent me, and I wanted to calculate these things to get an estimate of the dimensionality in that 8-dimensional trait space that was measured. If we don't assume multivariate normality, is there some lower than 8-dimensional space that things are operating in? And so the data look like this. Here are the 8 trait values that I mentioned earlier, and there's 124, so these are breeding value estimates. There's 124 of them. And what I need to do is set this resolution of selection. Remember, that was the width of the bin that I'm selecting for. With 8 traits in a data set of 124, there's really not much more you can do statistically than choose 2 bins per trait. So you can think of that, my resolution of selection, as being one where I'm either imagining selecting upward on CHC1 or downward on CHC1. Same thing for all the other CHCs. And the question is, how independently can I select upward or downward on these 8 different traits? How many dimensions really are there? So I can calculate the joint entropy of this data set. What I need to know then is a null distribution, like Mark talked about in his examples under multivariate normality. You want a null distribution for what you'd expect to happen if there really was no dependency among the traits. And so in this case, the way I got this null distribution is I just took random permutations of each of these columns. And what that means is what I'm effectively doing is reconstructing individuals, where I construct the first individual by choosing one of the CHC1 values at random, choose another one at random, and all the way through I construct an individual and do that again until I end up with 124 individuals. And now there should be on average, no dependency among those variables. And then I can calculate the joint entropy of that. If they're independent, the joint entropy should be the sum of the marginal entropies. That's the biggest I could expect this information entropy to be. If there's any dependencies, I'd expect it to be smaller. And so I do that a bunch of times, and I get a distribution, which is the null distribution under independence. If these traits really were independent, the joint entropy should follow this distribution here. And if you calculate the joint entropy of the data, you can see that it lies significantly below that null distribution. And what that means is it's extremely unlikely to get a data set that looks like this if the traits really are independent. There's clearly some dependency here among these traits, regardless of what the distribution is. We don't need to say what the underlying distribution is. Then what you could do is you could ask the same question, well, maybe there's only 7 traits. I could generate a null distribution when there's only 7 traits. Does the measured joint entropy fall outside that distribution? And by stepwise going through those, you would reach a point where you can no longer reject that possibility, and that would tell you something about the dimensionality of the space. Another thing you can do, which is what I'll show you here, is you could look at the joint entropy of the data and divide that. So when I calculate one of these random permutations, that's going to give me a data set under the assumption of independence. I could calculate the joint entropy of that, and I'd expect that to be bigger than what I would get in the data, because the data really do display some dependencies. So I'm going to get some fraction here, and when I multiply that by 8, that's my estimate of the dimensionality. And I do that over and over again with these 1,000 random permutations here, and I get a distribution for the estimate of the dimensionality of the genetic space. And that's what this distribution is here. And you can see this distribution is centered somewhere around 6.9 or 7, which, at least in terms of this data set, suggests that, again, focusing on a selection resolution that has bins of 2, there's somewhere in the neighborhood of 7 dimensions in this data set as opposed to 8. So there is one dimension that is missing in the sense. There's a direction on average, in some sense, that has got no genetic variation in it. So I'll conclude there just with these 2 summary points, that information entropy is in many ways a natural and kind of general way to measure evolvability and constraint when we're not restricted to multivariate normal. And aspects of mutual information provide a natural way that can be used to detect dependencies among random variables in general, and so naturally dependencies among traits and things like data involving genetic variances and covariances. And again, thanks to Mark for the data and all of these people who